Now let's look at purine synthesis. The purine base structure is shown here schematically. It consists of two ring structures. The purine bases are larger than the pyrimidine bases. This schematic summarizes the synthesis of purines. As shown on the previous slide, we can see that the generation of PRPP is required. Technically, this is not considered a step of purine synthesis, and the first step in purine synthesis starts here with the condensation of glutamine with PRPP. In purine synthesis, the 5-carbon sugar starts as a substrate in which the ring structure is built onto. From there are a series of reactions summarized here where the ring structure is built by the addition of each of these compounds. This pathway converges on the synthesis of inosine monophosphate, or IMP. Keep in mind, the base on inosine monophosphate is hypoxanthine. From there, inosine monophosphate can be converted to AMP or GMP. AMP synthesis requires GDP, and GMP synthesis requires ATP. We'll look at this a little more closely on a few slides. From there, the diphosphate forms, ADP or GDP, can either be used directly for RNA synthesis once converted to the triphosphate form, or can be accepted by the ribonucleotide reductase, denoted here as RRR, be converted to the deoxy forms, DATP or GDTP, and used for DNA synthesis depending on the needs of the cell. Let's look closely at the regulation of the first step in purine synthesis. The enzyme involved here is glutamine phosphoribosyl aminotransferase, or GPRT. This is the first committed step in purine synthesis. Here you have PRPP being aminated here by the addition of an amine group from glutamine. This enzyme is inhibited by the presence of both GMP and AMP. Both GMP and AMP must be present at high levels to inhibit the enzyme. This is because both GMP and AMP are synthesized separately in downstream parts of the pathway. Additionally, this enzyme is activated by the presence of PRPP. This slide summarizes purine synthesis and the regulation at various levels. Here we have the synthesis of PRPP. PRPP synthetase, as we've said, is inhibited by purine bases, shown here. The second step, glutamine phosphoribosyl aminotransferase, is also inhibited by high levels of both purine bases. The pathway converges on the presence of IMP. IMP can then be synthesized to GMP, or it can be synthesized to AMP. The synthesis of AMP requires GTP. Likewise, the synthesis of GMP requires ATP. Additionally, high levels of GMP can inhibit its own production, and high levels of AMP can inhibit its own production. This pathway is a good example of negative feedback, where a pathway regulates itself through product inhibition. The intermediary base in this pathway, inosine monophosphate, is shown here. It contains the base hypoxanthine. This base is not found in DNA, but is important when we start to talk about tRNAs or transfer RNAs. The addition of this base on the anticodon of tRNAs is critical for wobble base pairing. This is something to keep in mind in later modules when we discuss translation. Purines are large bicyclic molecules, and they cannot be fully cleaved or broken down within the human body. Consequently, they are salvaged or recycled to conserve energy. Likewise, these nucleosides can come from your diet, and those can also contribute to the pool of purines. Purine bases are large bicyclic molecules, and they cannot be fully broken down within the human body. Consequently, to conserve energy, there are salvage pathways that allow for the recycling of purines. Additionally, dietary intake of bases can also contribute to the purine pool. There are 3 classes of enzymes involved that are integral to the salvage of these bases. The first is a phosphorylase. This cleaves IMP and GMP to their free bases. Those free bases, remember, are hypoxanthine and guanine. The second class of enzymes is a phosphoribosyl transferase. This transferase allows for the transfer of a ribose 5-phosphate or sugar group to a free base. This reconstitutes a nucleotide. The last class of enzymes is a deaminase. This allows for the recycling of adenine bases, where adenosine and AMP can be converted to inosine and inosine monophosphate. Adenosine is the only base that can be directly phosphorylated. Keep in mind, adenosine consists of adenine and a 5-carbon sugar. Again, adenosine is the only base to be directly phosphorylated to reconstitute a full nucleotide. These salvage pathways are shown here. Important to the salvage of IMP and GMP is an enzyme HGPRT, here and here. This enzyme is a ribotransferase responsible for adding a 5-carbon sugar, PRPP, back to a free base, hypoxanthine or guanine. In the recycling of AMP, shown here, remember adenosine can either be deaminated to inosine, or this base can be directly phosphorylated by adenosine kinase to generate AMP. AMP can be deaminated to form IMP. This interrelationship of the nucleotide salvage is important, and dysfunction of the pathway can lead to pathological disorders. The most important step in this pathway is HGRP, shown here, which again is responsible for adding a 5-carbon sugar back onto the free bases hypoxanthine and guanine to generate IMP and GMP. Let's look at why that step is so important. In the degradation of purines, this converges on the generation of a compound, xanthine, by xanthine oxidase, the enzyme shown here. Xanthine can be converted to uric acid, also by xanthine oxidase. Uric acid is insoluble. Consequently, accumulation of it can cause a situation termed gout, which usually occurs by the accumulation of uric acid in joints, specifically in the large toe. This causes inflammation, which is very painful. Remember, guanine can be recycled back to GMP, and hypoxanthine can be recycled back to IMP via the enzyme HGPRT. This is important. Loss of HDRP and loss of recycling ability can lead to the overproduction of xanthine and the overproduction of uric acid and lead to a pathological condition of gout. The other situation where gout can more commonly occur is by an underexcretion of uric acid. This is usually occurs because of a poor diet or other compounding situations that lead to underexcretion of uric acid and gout. Allopurinol, shown here, is a pharmacological agent that can inhibit xanthine oxidase and can be used as a treatment for gout. Let's look at this question. Gout can be caused by elevated levels of uric acid, which we just saw on the previous slide. With respect to a possible HGPRT defect, which is most likely to cause gout? A mutation leading to decreased activity of the enzyme, a mutation leading to increased activity of the enzyme, or a mutation of this gene is unlikely to affect the occurrence of gout. The correct answer here is A. Decreased activity of HGPRT can lead to a decrease in the recycling of guanine and inosine, contributing to gout. This is summarized again here. HGPRT is responsible for the conversion of guanine to GMP and hypoxanthine to inosine. Decreased activity causes increased xanthine and hypoxanthine production. Severe loss of this enzyme causes Lesch-Nyhan syndrome. In addition to gout, this causes severe mental retardation as well as compulsive self-mutilation. Partial defects of this enzyme can cause premature gout, but again, none are as severe as LNS. This is rather uncommon, and the most common cause of gout is underexcretion of uric acid.